afternoon, early evening for you guys in India. So this is really fun to be here. I'm currently assistant professor in the University of South Florida in Tampa. So we're in the land of manatees and alligators. That's our biggest wildlife. We actually have an alligator just outside our house right here, but he's not invited to the meeting this morning. He's just hanging out outside. I'm so excited. Shreya and Karishma invited me to talk to you about visualizing microbes. And that's kind of what we spend a lot of time doing in my lab. So I have 3 or 4 students that work with me. And we spend a lot of time looking at microbes and trying to understand how they interact with each other, how they can resist antibiotics. So that kind of fits in nicely to your next session. I think you'll see some overlapping themes. But this is just like a little artistic version of, a lot of agar plates with microbes on that you might see in the lab. And I'm going to talk to you a little bit about microscopy because that's what we do a lot about in our lab. Okay. So I always like to kind of just start off with, what is microbiology? Of course, you're in these sessions, and so you're interested in microbes, but just to kind of remind you how I see microbiology in terms of it's the study of a large and diverse group of microscopic organisms. So that's the really important part for what we're thinking about here today. And really importantly, they can exist as either single cells or clusters, and that's something that's going to be a theme that we're going to talk a little bit about. And so in terms of research, when we think about microbiology, it's not just about disease and medicine. Of course, medicine and infectious disease are really important. That's what we do focus on in the lab. But I think using microscopy, it's important to know that you can use it in lots of other aspects of research. So agriculture and food sciences, immunology, ecology, genetics, and many, many more. And I guess the important thing is to realize is that not all microbes are bad. And so if you're interested in science at this stage, maybe at some point microscopy might feed into some of these other areas that you might end up working or studying in. So if we think about microbes, important for medicines, foods like cheese. I mean, I think cheese is one of my favorite things in the world, and that's really, a big part of microbial life. And beer. You're all a little too young for that right now, but at some point you may enjoy that. And of course, bacteria are living organisms. I think that people kind of forget. We hear all this bad reputation of them. They cause disease, but they are living organisms. And so like us, they like to interact. And that's something we're going to talk about in terms of how actual bacterial cells interact with each other. They reproduce like all other living organisms. And they need nutrition like all of us. They have their favorite foods, they have their favorite places they hang out and get those foods. And those are all aspects of bacteria that we can study, and we can visualize them doing these different aspects of their life cycle. And then just a little reminder as we're kind of looking at these different microscope images and talking about looking at them. I'm sure you've probably touched on this before, but bacteria are very diverse in terms of what they actually look like. So if we think about different bacteria that we may see, the ones that I study are here, this bacillus or rod-shaped bacteria. They can be round, spherical, cocci bacteria, but there's also some really cool ones that are kind of spirals or what we would call spirochetes that you might find in the oral cavity. So I'm sure at some point, if you're having these guest speakers in your sessions, you might come across some of these really cool different shapes of bacteria. So that's really something to kind of keep in mind. Should we stop for a couple of questions just before for this little intro part?
Sure, Sophie, we can. So right up front, Kavish, who's a regular Talk to a Scientist participant, asked a question. What do you mean by visualization?
So visualization is really how do we see these microbes. So of course, to the naked eye, if you're just looking at something on an agar plate, you can see if we grow bacteria in the lab, you might see a colorful agar plate, which is just that kind of jelly material that we put bacteria on as standard. You might see a lot of different colors, so we can see that something is growing, but by the naked eye, we can't see individual cells. And so when we talk about visualization, we're talking about ways that we can actually look at those individual cells and see what they're doing. And microscopy is one of the easiest ways to do that. So I'm going to talk to you a little bit about some light microscopy that I use, but there's lots and lots of different ways you can use microscopes to look at things that you can't normally see just by the naked eye. Hopefully that answers your question.
Yep. We look forward to it. And Tavish, follow up with another question if you have if you have any, if you have any questions. Kavish asks another question, actually, he does, Sophie. Does shape affect the working or structure of the bacteria?
Yeah, definitely. So shape is something that's really important. So I think a really good example of that would be if we're comparing maybe these cocci bacteria, maybe with the bacteria that we're going to talk about today. So you can't actually see it on these rod-shaped bacteria, but they have little tails, which we call flagella. That's their official scientific name, but their little tails actually help them swim. And so these bacteria are able to move from one place to another. Whilst these bacteria here, the round ones, are much more stationary. They stay in a place, they're not able to move around. So that's a really important thing about the shape of bacteria that really would make a difference in terms of how they work, how they may cause infections or cause resistance to antibiotics, etc. So that's something that's really important. It's a great question.
Absolutely, Sophie. Great question. And as you would see, some of these are actually open research questions. I mean, someone could earn a PhD for asking this question.
Yeah.
How does shape affect the working of a bacterium, right?
Yeah,
great
absolutely.
Question.
And then one more question just popped up. If you were looking through a microscope at a bacteria, won't they, the lens get infected? No. So that is definitely something to think about. So when we're looking through the microscope, there is a distance between your sample, so where your bacteria is and where you're actually looking at it. So you are protected for sure, but there is a working distance between that. So you're definitely protected from that. But of course, putting your sample together, if you guys ever spend time in the lab, there are certain ways to handle bacteria to keep yourself safe and protected. So that's something you'll work on if you're in a lab environment, but you're certainly safe looking through a microscope.
Sophie, there's one more question from Kavish, and after that we'll move on and come back to these questions. What about fold scope? Oh, Ala also has a question. Okay, we'll take two questions then. What about fold scope? There is very less distance. He says, like maybe you can have a working distance in a light microscope, but in the fold scope, could we?
I'm not sure what a fold scope is. Am I allowed to say that? I've never heard of that before.
Sure. You're absolutely. So it's something that it's sort of like Manu Prakash in Stanford came up with this paper microscope with a lens, which you could use to sort of look at river bacteria or larger bacteria in rivers and things like that. But there too, Kavish, just to answer the question, you can keep some distance there too. Yeah. So it's like a mobile microscope, basically, Sophie.
Yeah. I see what you mean. Yeah, I think I remember having one of those at one point. Yeah, I mean, obviously, for the physics of it, which we're obviously not going to get into today, you definitely always have to have some distance. So even though you might not think there's a distance, there is in terms of what's protecting you from that.
Okay, we have more questions, Sophie. Do you want to take them? Should we come back to them?
Let's go on a few more slides because I think it might answer some of them.
Okay, sure. Okay. Ala, Arshia, and Tiago will come back in a few slides.
Let me just go back. Okay, so we talked a little bit about shape. Had some great questions come up there. And then I guess another thing that kind of leads on to shape. So this slide here shows you, again, those different shapes. You'll see some here, that spiral shaped bacteria again, the cocci, these bacilli. Here again, you can see these little things coming off of the bacilli, and that's what we would, the bacteria may use to attach to a surface. But I wanted to use this slide to show you, this is supposed to represent the inside of your tummy, so the inside of your gut. And you can see what we end up with are these very diverse mixed populations of bacteria. So if we kind of correlate these little colorful houses to different bacteria, you can see if you're going along your gut, you actually get these very diverse groups of cells that live together. So bacteria like to organize themselves in these groups, very diverse groups, kind of like the diverse group that's watching this session. And what we're going to talk about is why these bacteria kind of organize themselves in this way. And when we talk about bacteria in the gut, I think that's something to think about, that they are a very beneficial thing for us to have in our gut. And that also correlates into this slide too, because I know some of you touched on good and bad microbes in the questions. And one of the biggest things, of course, that we're talking about, they can either live as small groups or clusters of cells. And this is really where we talk about bacteria growing as biofilms. And so here again, we're representing, it doesn't have to be a single type of bacteria. You have a few of them here grouped together. They can attach to a surface. they grow. So they're either reproducing and taking in nutrition, like we said, and interacting, like in that, the fact that they're living organisms and they have all those different aspects to them, like we do as living organisms. And then what happens is these biofilms, they disperse. So a couple of cells will come out of that little community, and then they'll go on to form new biofilms here. So when we're talking about biofilms and how we might think about looking at these bacteria growing over time, this is something that we're looking for all the time as scientists, to look at these structures develop and kind of understand how you get from one stage to another. And that's something that's really important in thinking about, well, if we wanted to treat an infection or treat something that's causing disease in a human or another host, how can we as scientists interfere at these different stages? And that's what we spend a lot of time thinking about, how we can interact with those stages. Let's just have a quick look at the question and see if something comes up. See that you can see the color when you grow bacteria in a Petri dish. So that was definitely a little overrepresentation of some of the colors, but bacteria are very colorful. So I'm looking at the question from Ashia, is that how I say your name? In terms of thinking, different colors. So Pseudomonas, the bacteria that I work on, is very, very green. And so when you grow that on a plate, it's really distinctive that it looks like it. You can sometimes, if you've looked at this bacteria for a long time in your life, and that's what you've built your career on, you are able to recognize it a little quicker than others. The other way to recognize bacteria is smell. Again, you have to have a little bit of a custom nose to that, but there are certain smells that are relevant to certain bacteria for sure. There is a bacteria called Klebsiella, which is very, very pink and sticky, and that's another one I think visually obvious which one that one is. So there are definitely ways to look at it, just how it grows on a plate for sure. Are there any ways to collect bacteria?
Ala asked a question. Yeah. Ala's question. Also, you can take them. Yeah, sure.
Oh, do the good bacteria last longer than the harmful bad ones? Last longer. I think the main kind of take home from that question is that we have a lot of good bacteria in places like our gut and our skin, and they can be there for a very long time. Harmful bad ones, we hope they don't last too long because we hopefully treat them with antibiotics and things like that. But there are some times in some conditions where people get infections for a very long time. And that's actually something that we study in my lab, are people who have lung infections. People with cystic fibrosis have lung infections that last for many, many years. But of course, good bacteria are there all the time. Hopefully that answers that question.
We can come back to it. No worries. Yes, Sophie. And then Tiago asks, are there ways to collect bacteria other than growing them?
There are. So actually, one of the biggest problems in science is that sometimes we don't have ways to grow certain bacteria. And this can be either from, if you're taking it from a human, or even if you're taking it from the environment, like from rivers or from the surface of a rock, for example. We don't always know what bacteria like to eat and thrive in growing. So what you can do is you can collect the sample just by taking like a scraping or a swab. And what we can actually do is use a lot of these new techniques where we can actually take the DNA and sequence that, and that tells us which bacteria are there. And that's a really nice way now to actually be able to see which bacteria are there versus, having a group of organisms, something doesn't grow and then we don't really know it exists. So there's using all these really modern DNA sequencing methods is a great tool now for us to understand which bacteria, like the unculturables as we call them, what's actually there in that site.
In fact, unculturable sounds like the name of a rock band.
I know. I feel like if I was going to be one, I would be the Unculturables.
Totally, yeah.
I'd play babes in the Unculturables. That would be great.
I know. Sophie, we'll take one or two more questions. Avishya has. This is a nice question, actually. Do bacteria have favorites? Like they prefer to be with some and don't want to stay with them?
Absolutely. And that's definitely part of their social aspects. So if we think about that in terms of bacteria that you might find in your gut, there's definitely a subset of organisms that like to be together. You'll only find certain organisms. Some of that is due to, they might have friends that all of them can resist a very acidic environment, and that's why you'll find them there. When we think of infection, there are a couple of instances where you have certain bacteria that we always fine together. So I mentioned the cystic fibrosis lung, which is what we're interested in. And we always find Pseudomonas and Staph aureus together in those environments, and a couple other ones. So definitely bacteria have favorites. Some of it's because they can benefit from each other. And sometimes it's one can take advantage of the other also. So in terms of humans, they're similar to humans. They definitely have favorites. I like that question.
Nice question. Okay, Sophie, we'll move on a little bit, get into the actual visualization part. We'll come back for these questions.
Yeah. Okay. And so thinking about biofilms, where can we find these biofilms that we just talked about? So we kind of touched on this a little bit. We think about in nature, environment. So if you look on the surface of rocks, that slimy substance, that's a biofilm. In the ocean here, you can even take biofilms off of different ocean plants or coral mucus, things like that. They all have microbes that are living there. Gross but true in our sinks and drains at home. So that nice scummy liquidy stuff, that's also a biofilm. And then our teeth, I think this is one that always kind of goes a little kind of discussed sometimes, but a huge area of biofilm research is thinking about when you get plaque, if you don't brush your teeth properly, that's a bacterial biofilm that's really interesting to study and definitely look at under the microscope as well. So ultimately, I mean, we've talked about biofilms, how they, you'll find them in all these different places, but it's important to remember, I'm going to come back to my old favorite, the cheese, not all biofilms are bad. So this is a nice artistic kind of representation of maybe microbes in your gut here or on your skin where you can see all these different species. So again, those different shapes of bacteria. And they're actually doing a lot of good for you by staying in your gut and keeping you healthy. Cheese also keeps you healthy, but obviously it's a nice addition to microbial growth and how we use microbes in everyday life to be really quite beneficial. Of course, not all bacteria are good, and the bacteria that we actually study is called Pseudomonas aeruginosa. We'll just call it Pseudomonas today, just to keep it short. But I don't know if you've looked at the website Giant Microbes, you can actually get a slightly enlarged version of your favorite bacteria. This is Pseudomonas. Those flagella that I mentioned. This is a good example of that here on Pseudomonas, and it helps it be very motile in its human host, or if you find it in soil or water. This is a really lovely microscopy image. This is actually scanning electron microscopy, which allows you to get such a really wonderful up-close view of Pseudomonas. You can see the shape of the bacteria here, and these flagella that are so lovely put on this little fairy microbe, you can see here in green, that's the actual flagella of the bacteria that would allow it to swim, which is really nice and a really nice way to visualize it. And as I said, Pseudomonas isn't just something that causes infections. You see it in the you can isolate it from soil. It can live quite happily on the surface of plant leaves. It's important to know that if you're relatively healthy, Pseudomonas isn't something that you're going to be interacting with as a human. Really, it's just something that causes infections in those who are a little immunocompromised or unwell to begin with. So as a bug, we really are interested in kind of understanding how it survives in certain human infections in our lab, but it is something that we find in many places in the environment. So normally in healthy individuals, this really is a clinical problem. And so we talked about visualization. So this is a really good way to show an example of visualization. So how we can grow them in the lab. So basically we can get bacteria to express or show themselves by expressing fluorescent proteins. So this one here has been labeled to turn green when it's been growing. And this is a really nice example of biofilm forming bacteria. So you can see these very kind of distinct mushroom shaped structures that develop over time. And so you can see here, this is using confocal laser scanning microscopy, which is what we use a lot in our lab, an example of a growing biofilm. So everything that's green here is a bacterial cell. You can see you're not able at this point to see individual cells. What you're seeing is a huge group or a coordinated group of bacteria that are stuck together on this surface and then developing and growing to form these big structures. So this is a really good example of how we might visualize bacteria in the lab. And you can imagine if you're maybe interested in seeing how these bacteria are treated with an antibiotic, you could add an antibiotic or something like that into the system and then keep looking at it and seeing how these shapes and structures might change. And then I think we'll just show this one and we can look up some more questions, but talking about looking at bacteria a little bit more closely, I mean, that was looking at something in the lab, but if we take an actual human infection, what do bacteria look like in that setting? And this is something that my lab has focused on now in terms of thinking, well, if we actually have a human infection, what do those bacteria look like? And so there's some good examples of, this is taken from a CF, a cystic fibrosis lung, so a little piece of lung tissue. And here in red are the actual Pseudomonas that we talked about. So this has just been stained and then looked at under a regular light microscope or confocal microscope. And you can see here the bacteria growing in these little micro communities, similar to that cartoon I showed you. And then this is thinking about what bacteria might look like in a chronic wound. So we talked a little bit, oh, do they have certain friends that they like to hang out with or certain partners? This is a great example of Pseudomonas and Staph aureus that grow together in a chronic wound. And this is oh wait, it's going to a video. Hopefully there we go. I don't know why I went to that, but there we go. Hopefully you guys can see that. This is just showing you how we can visualize Pseudomonas and Staph together in a wound. So this is a nice example of that. There we go. So maybe a couple more questions. I see 30 new messages, so
yeah, there are lots of questions, Sophie. Sure, Sophie. Let's start right at the top. Arshia has a very nice question. So hand washing soaps as 90%, kill, their marketing line is we kill 90% of bacteria. Are they also killing good bacteria? Is the 10% probably the good bacteria that they don't kill?
I think there's definitely a chance that you are getting rid of some of the good bacteria a little bit, but ultimately washing hands is killing the bad bacteria that we don't really want to be passing on to other people as well, or putting onto touching surfaces. I think the great thing about good bacteria, when we think about in your gut or on your skin, especially on your skin, because hand washing has been such a topic the past couple of years, those bacteria are very readily able to replenish themselves. So keeping that nice, healthy balance on your skin, that's their goal really, is microbes, is to kind of keep maintaining that protective layer or beneficial layer. So certainly you're killing off the bad ones by washing your hands, but your microbes know what they're doing and they keep you protected too at the same time.
Thank you, Sophie. Thank you. So Arshia asked another question later in the chat. How do you take pictures? How do you take these pictures from a living organism?
So a living organism. So when we're actually looking at just the bacteria in the lab, we actually grow them in a special media that has all of the nutrients and all the food sources that they really like. And so what we're able to do is take pictures very, very quickly. So I said that, the bacteria can move. What we do is we take images very, very quickly, like every second or so, and then we're able to actually capture those bacteria actually moving. In other settings in science, so some of the examples, like the ones I just showed you, that would be what we call a fixed sample. So something that's been taken from a source, you have a tissue, and then you're staining it and visualizing it that way. Other ways of visualizing microbes, and maybe if you had a mouse model, you wouldn't be able to see it at this high resolution. So you wouldn't be able to see single cells, but you can see an accumulation of light or fluorescence or something that you've labeled your bacteria with, which you can actually visualize in real time. But again, that would be more of a collection of light versus something single cell level.
All right, so we can fix them. Okay. Avishya says, can you change this is the last question for now and we'll come back later. Can you change the DNA of a bacterium, like make them bad to good?
We can certainly manipulate or change the DNA of bacteria. We can add in different things. We can take things out for sure. So one of the big parts of science is what we call making mutants. So if we're really interested in something that's in the DNA of a bacteria and we think it might be beneficial or it might be something that helps the bacteria cause disease, we can study that by knocking it out or deleting it from their genome. So we can definitely manipulate bacteria. The goal ultimately, absolutely, is to be able to do that to maybe prevent disease. A really good example of this is, I'm not sure if you guys would have heard of the bubonic plague. So that's something called Yersinia pestis, when we look at that in the lab, that's had the main toxin or the main part that causes disease knocked out, so we can actually study it in the lab. So that's definitely an example of making it from bad to good, at least so we can study it. But I think that that kind of method is definitely something as scientists, it's going to be really important for us to actually, have really good methods to treat these infections. So that's a really great question.
Right. All right, Sophie. Thank you. We'll move on and come back.
Okay. And so in our lab, I talked to you a little bit about cystic fibrosis. So I don't want to go too much into the science of that, but really cystic fibrosis is something that a subset of people are born with it. It's a genetic disorder. So there's a lot of children who suffer from infections of Pseudomonas aeruginosa. And ultimately, what we're trying to do in the lab is simulate an environment that's very similar to what might be happening in a patient. And so Pseudomonas causes infections in the lung. And so what we're trying to do in the lab is simulate or mimic an environment very similar to the lung. And so what we do in our lab is we grow something, we grow our bacteria in what we call an artificial sputum. So sputum really is just the gooey, nasty stuff that builds up in the lungs in these patients. We try to do bacteria in it. So that's what we use in our lab primarily. And how do we visualize them? When we use this confocal microscope. It's a really nice instrument that we get to use in the lab every day. And it's probably my favorite part of my job. I think it's one of the most amazing things. A lot of you had in that word cloud, mind-blowing. I think the first time I ever used it, that was probably one of the adjectives I had. And it's really cool as a scientist to be able to go to work and have access to tools like this so I can study things every day that I'm really, really interested in. And so this is kind of one of the images that we might create in our lab using that mimicking sputum, our lung environment and the bacteria that we add into it. And we're using that microscope that I just showed you. And so here, what we're actually looking at is our bug Pseudomonas in that very sticky, viscous sputum media. And you can see, we can look at it expressing green fluorescence, and we can actually see these bacteria growing in this environment. And we basically just use what looks like a regular microscope slide that you might use in the lab. And it just has these little wells, so we can look at a little bit more volume. So that liquid sputum that I showed you, we add our bacteria in here, and then we can look at them on the microscope, and we get lovely images like this. And then what we do in our lab is we use a lot of computer technology, imaging analysis to really look at these little individual groups. So this is basically taking one of these out so we can look at it as an individual. And why is this useful? I mean, yes, it's fun to grow things in the lab and look at them, but I think really we always have to look back at the bigger picture as scientists and why we're doing this. So this is a really cool tool to really start asking questions about how maybe we can study antibiotics. So if this bacteria, this little group of bacteria is resistant to to antibiotics, can we understand why that's happening? So we can add antibiotics to those little biofilms that I showed you. In the host, we can add immune cells. So you might talk a little bit about immunology in your sessions here. So your host cells that you produce in your immune system, how do they interact with this little group of bacteria? We talked about favorite partners or friends that they might have. How do they interact with other microbes? So we can add in lots of different other microbes. There's different shapes, sizes, species. And then something that you might hear about if you're reading different science articles or things in the news, bacteriophage. So this idea of viruses that infect bacteria as a potential way to treat infections other than antibiotics. So how do they interact with these little groups of cells as well? And that's something that we can also study in the lab. So that's something that we're working towards always. We may want to answer a couple questions before we move on to the conversations part. I don't know how we're doing on time.
We're good, Sophie. It's 5:39. I mean, I don't know what time it is for you, but we have 20 minutes left. Yeah, okay. We're good. We'll take one question, Sophie. Yeah. Arshia again asks, isn't it unsafe to study dangerous bacteria up close?
Everyone's very into safety. I like this. I wish some of my students were so aware of this. Safety, yeah, absolutely. It can be a threat to you as a scientist to look at certain microbes at a very close distance. But in labs, what we actually have are a lot of different mechanisms to protect ourselves. So when we have different bacteria, they're actually grouped into levels of kind of the threat that they are to you to actually study them. And that's called a biosafety level number that we get given. So most bacteria that we actually study in the lab are what we call biosafety level 1 or 2. Biosafety level 3 and 4, which you might actually always hear about on the news. So COVID, when we talked about COVID-19, was BSL 3. And when you look at those microbes, if you're looking them under microscopes or just handling them in general, there's special facilities that you actually work with those microbes in. And I'm sure there's a really great opportunity to Google and kind of find out about those BSL 3 and 4 facilities. And they're not everywhere. They're a little bit more scarce than what we do in regular labs, but there's certain ways to protect yourself if you want to study those as a scientist. And, they're very, very effective in keeping you safe.
All right. Okay, that's great. Look at the questions. It leads us to different directions. Okay, we'll go on. Yeah, Sophie, we'll go on.
Okay. So one of the things I've been talking about, we're visualizing bacteria. We talked about the different ways that maybe we might be able to use it as a tool to look at things like antibiotics and how they may interact with our host immune cells. But one of the things I've always, how I was introduced to science and why I really wanted to stay in the science field is really thinking about how bacteria are very, very social. And so the idea that we like to have conversations or we interact with our friends as living organisms, this is something that we can actually look at and observe in bacteria. And this is always one of my favorite things to show that, this is something that we've seen and we've known about in animals for a very, very long time. And a really great example of this is meerkats. So meerkats are a really wonderful example in the animal kingdom of organisms that interact with each other. So they normally live in these groups and they come out during a certain part of the day to hunt for food. And they're very, very good at signaling to each other to know when it's safe for the rest of the group to come out of hiding and when also to retreat back to their safe place. And I think they were a great example and also very cute to show that this is something we know about in animals, but we haven't really looked until very recently or the last 20, 25 years as to how bacteria actually interact. And it's very similar in terms of they work as a group to interact with themselves and with the environment that they're in. And this is something that we call quorum sensing in microbiology. And it's basically, if anything you take away today from the session is it's how bacteria socialize. So I thought this is a great cartoon. I just only found it the other day, but this idea that they're all holding light bulbs, basically. When they're really close together, they're able to interact and we get all this light. If you're out on the outskirts here, you're not really able to interact with the others. And so your little light bulb isn't on. And I think this is a great example to show you in a cartoon of how bacteria kind of interact with each other. And this example of, light is something we can actually see in the lab. So this is an example of a marine bacteria. It's called Vibrio fischeri. And basically, if you grow it to a really, really high density in the lab, this is in a culture flask, and it's in a dark room, you can actually see that it makes this very, very vivid fluorescence, which you can see. This is just by the naked eye as well. So this is if you have a culture or a big flask of cells, you can see this wonderful bioluminescence actually occurring here. And this is just a little schematic to show you. So if you had just the media that you were growing your bacteria and you put them in at very low density, so when there's just a few cells, so this would be here on this outskirts, if you just have these 4 cells maybe potentially hanging out, when you get them reproducing in this very dense kind of buildup of cells, this is when we get this bioluminescence. And this is what you see here in these culture flasks. So this is a great example of how these bacteria are interacting and basically producing light in this case. And where do we see this in nature? So I talked about Vibrio fischeri. Vibrio fischeri is an organism we find in the ocean. And so there's one organism that uses this bacteria as a really useful tool, and that is this Jamaican bobtail squid. So he looks like a very funny guy. He's hanging out. He also is a living organism. He needs to eat and he's a predator. He has food that he likes to eat. And he actually uses that bacteria that luminesces as a really useful tool. So it actually has this bacteria living inside an organ in the squid. And so what it does is it has very low levels during the day of this bacteria. During the day, the bacteria divide and reproduce and get to that high cell density. And then at night, the bobtail squid uses that light to actually find prey when it's looking around in the ocean looking for prey at night. When it's finished feeding, it gets rid of all the bacteria and the whole process starts again the next day. And so it's a really useful tool that the actual bobtail squid uses and utilizes from the bacteria that lives in its actual light organ. A more modern reference to this of how this might actually work, and maybe many of you have seen Finding Nemo, but that's a really good example of actually bioluminescent bacteria to kind of give you this concept a little bit more in a modern context. And why do bacteria do it? And it's all about strength in numbers. So obviously those bacteria are working together to produce light. That's been really important for the bobtail squid. But how do we think about this in terms of human infection? Really, it's about those cells working together to do something that causes disease. And it's really about strength in numbers. And I always try and think of a good way to show this in terms of, what do I mean by strength in numbers? Well, if you think about a supermarket, you have this supermarket aisle here. Imagine if you had one raccoon. So a raccoon loose in the supermarket, it's a nuisance, but it's not something you can't handle. But imagine if you had many raccoons in one supermarket. That's when you've got strength in numbers. These raccoons are going to cause absolute mayhem. And that's really, I always think about in terms of bacteria do much better as a group than they do when they're by themselves. And then if we think about that in terms of humans and how we may cause disease and infection, we talked about, they make light. That's one example of why they work together in the quorum sense, but also things like antibiotic resistance, which you're going to talk about in your next session, that's linked to them working together as a coordinated group. Forming biofilms, like I've shown you there, they can form biofilms. That's a coordinated effort. Movement, we talked about flagella. So that's, again, something that can be related to them working together as a group. And then just things like digesting food. So when they're living in a certain environment, they can work together to do that. And then thinking about disease producing toxins or things that might cause disease is something we'll actually do is working together as a group. I don't know if we have a couple questions we can do, and then I think it's just a couple more slides.
Okay, we'll start. So Ala had a question. So quorum sensing is related to bioluminescence, right, Sophie?
Yeah. Yeah, okay, absolutely. Yeah. So actually the best way that we can answer this question actually with this slide. So I didn't really want to put too much of the actual molecular aspects of it, but there's a really simple way to describe how quorum sensing and bioluminescence are together. So I talked about you have a few cells, you don't get any light. If you have a lot of cells, this light is produced. And it's all based on the fact that bacteria are able to produce little signal molecules, and they're also able to sense them. And so this schematic here tries to show you that all bacteria can produce these little signals. They can recognize them, so they'll have little receptors on their surface that allow them to sense these signals, and then they'll act accordingly. So either light, virulence, something like antibiotic resistance, or their form of biofilm. So quorum sensing basically is a way that bacteria coordinate efforts as a group. So bioluminescence is one, but it could be something else like producing a toxin or working together to digest a food. So maybe they'll all produce an enzyme to break down food. And then it's all basically the benefit then is for all of them as a group, and it makes them stronger. And that's that strength in numbers aspect.
Wonderful, Sophie. So Bhargavi continues that and says, how are the bacteria so bright?
How are they so
bright?
Bright as in bright light, or just clever bright?
No, bright light. I think she means bright light, but yeah.
Okay. They're so bright because there's so many of them. That's the thing. I mean, we might, whenever we look at kind of cartoons of bacteria, you see maybe like 20 or 30 cells, but we're talking about millions and millions of bacterial cells. So imagine you have that many bacterial cells all expressing this bioluminescence. You can see why it is such a bright light. So in that volume of a flask culture, that's maybe 1 liter of liquid. And within there's millions and millions and millions of bacterial cells all producing that light. And that's why it's so concentrated and so bright.
Wonderful, Sophie. Thank you. And Bhargavi follows up with another question. Where are all those bacteria in the squid? Where are they?
So they're in a part of the squid called the light organ. So it's basically on the underside of the squid. And the reason it's on the underside is actually so an extra aspect, which I didn't share in that slide, was that the squid actually uses some of the moonlight on its back to kind of cast a shadow of itself, and then also uses the bioluminescence from the bacteria. And the two of them work together to keep the squid from being detected by the prey that it's actually hunting. So there's another element to it, that the squid is like a super amazing, clever technique that it uses to actually find food. But the light organ is situated on the underside of the squid. So you can imagine squid shining light down on the bottom, maybe in or below it in the ocean, to actually find the food that it's trying to eat.
That's really cool. And that makes us rethink intelligence in creatures, right?
Absolutely.
Yeah. All right, we couldn't do that. So, okay, Sophie, Ala has a great question. I've heard of bioluminescence in sea algae, like squid, they have algal bioluminescence. How is bioluminescence helpful to the algae, or is it similar to squid bioluminescence? Algal bioluminescence?
Yeah, I mean, I think it's also whenever you see substance in nature, it's definitely something that's a benefit for them to either be detected or not detected, and also, to look for food, but also to sometimes just be a way to warn off predators too. So, if they're there as a group, as a mass, then it's also a signal to others that, we're here, don't mess with us, that kind of aspect too. So you can see it from good and bad perspectives, but algae will work in the same way. I'm not saying they work in the same way down to the molecular level with the molecules that are involved, but the overall concept is the same. They're using it as a benefit to kind of sense and understand their surrounding environment.
All right, Sophie, we'll move to the last slide, and there are lots more questions. We'll try to take them all.
So this idea of watching bacterial conversations, so this ability to interact with each other. So when they actually quorum sense, there are certain genes that we talked about. Can we interrupt the DNA? Can we change the DNA? Well, we know in the bacterial DNA that there are certain genes or certain stretches of nucleotides that are important for quorum sensing. And what we can do to study them in the lab is we can label those. So we can put a fluorescence protein in the DNA, and then it allows us to actually watch the bacteria start talking to each other. And so what we actually use is what we call a fluorescent reporter in the lab. And this video I put in to show you how we can see here in red, you'll see this is just a very dense or full bacterial culture in red. And then what you're going to see are little spots of green pop up. And where the green is where the bacteria are actually having conversations and talking to each other. So that's where they're quorum sensing. So let's see. Here we go. It's going to open up again in here. I'm going to just do a full screen so you guys can see it.
I am not able to see the video. Maybe you'll have to share that screen as well.
Oh, okay. Let me undo share. There we go.
No, Sophie, not yet. We just see we don't see a video.
Okay.
Yes, now we see the video. Yes.
Okay, perfect.
Absolutely. Great.
Yeah, so what you'll see is the red, which is all the bacteria, and then certain ones are green, and that means that some of this bacterial population are talking to each other. So even here you can see there's little single cells, there's groups of cells that are green, and this is basically how we can study those bacterial conversations. So we can actually identify which cells are having these conversations, which ones are quorum sensing and which ones are not. I'll just play it again so you can have a good idea. But you can see the biofilm almost looks like a little bit fluffy too. So we can kind of see, all the kind of extra things that bacteria have around them as a protection layer. So a matrix or such around them that protect them in the environment. But this is a really nice way to distinguish who's talking and who's not, really.
It's like when the color is like raising your hand to speak,
Yeah, absolutely.
I'm saying something. Yeah.
Absolutely.
Beautiful. Maybe we'll see the video once more, Sophie, if it's okay.
Yeah, let me go back to there. There we go. So also I should say, this is a video that you're seeing actually a lot of time squeezed into one small video. So the bacteria that we're actually looking at will divide every 30 to 45 minutes. So this is actually a lot of time condensed into one short video. So we'll probably take images maybe every 25 to 40 minutes so we can capture those divisions and seeing it. But that's a nice thing that you can speed up to actually show people, how this visually happens. But of course, this happens over a slightly longer period of time. That's important to remember.
Oh, absolutely. And we can see that our participants are getting it. The bacteria are talking, basically what Arshia is saying. Okay, Sophie, go ahead.
Yeah. And then that was my last slide in terms of things that actually show you, but we definitely have some time for some questions. This is just to show you that this is my lab. They are obviously real people, not cartoons, but they really like to have this version of us on whenever I have slides. You can see I have a few different people that work in my lab with me every day to kind of start looking at these questions. My dog Maggie is our honorary member because she's been with me since the beginning. But we're funded by these different people here that really help us kind of keep our science going. But if any of you have any questions afterwards, please feel free to email me. This is my work email address. And you can also follow us on Twitter, and that's our lab website too. So it'd be great to hear from some of you if you have more questions afterwards. But of course, we have questions now, I think, still in the chat.
Yes, Sophie, thanks for sharing your information, and we'll get in touch. Absolutely. Yes. So Bhargavi just wants to know once again, so why are some bacteria red and some green in that video? So the red ones were not talking.
Yeah. So in that example, the red ones were not talking. So what we need to do to actually look at them on that type of microscope is they all have to be at least labeled with one color so we can actually see where they are. And then what we do is we use other colors, which are maybe tagged to a gene, so a talking gene. And then when we know that talking gene is on, we can see the other color. That's one of the, I guess it's an advantage and a disadvantage. So if you look under your regular light microscope in the lab, you can see the bacteria and you can see them, they look gray and black, like a phase contrast, what we would call an image. So you can see the different bacteria, but when we're looking at them under that very much higher resolution where we can see single cells, everything has to be labeled with something so we can see it. And then what we're ultimately looking for is color changes to indicate something is happening. So like Karishma said, the green is I'm putting my hand up, I'm signaling to you, and that's how we look at the changes from red to green.
Wonderful, Sophie. It was a lovely session. It was really very high quality engagement.
I think, Mayor's question is, can we create our own bacteria?
Yes.
So I think we can definitely create versions of them. And that's ultimately a goal that we can, in the lab, we can create different versions, and that can be good or bad. But we can definitely manipulate them, which means we can change them. And that can be a really useful tool to actually understand them at a more molecular level. So I think, yes, we can definitely change them. Create them from scratch, not so much, unless we're just talking about we grow them in the lab and we create new ones from one. But ultimately, it's really about how we can change them and manipulate them to use them as tools to understand how they really cause infections. I think there's one more question.
Yes, Sophie. How do get people
get infected with Pseudomonas? Where do we contact them? So actually, as healthy individuals, we will be exposed to them. They're in the environment, they're in soil, they're in water. But if you're relatively healthy, it's not really going to impact you. You're going to have it, your body's going to get rid of it if it wouldn't cause any disease. Really, what we worry about are a certain group of patients. So we have people that study cystic fibrosis, people who have wounds. This is people maybe who have like diabetes or chronic underlying conditions. Those are really those who are affected by Pseudomonas. So not something as a healthy person you really need to worry about. But obviously, we are worried about those people that do get it, and that's why we study it and why we're interested to see how we can target it.
And so with one final question, with 3 minutes over time, but just one question from Arshia.
Viruses kill bacteria and reproduce, but can we manipulate bacteria to kill the virus? We can manipulate the bacteria to become more resistant to those viruses, and some bacteria are. So when we talked about bacteriophage or phage therapies, they're viruses that are very specific to target a bacterial cell. Like bacteria evolve resistance to antibiotics. They can also, unfortunately, be resistant to these viruses as well. And so, trying to understand that is it's a very modern part of science, really using them as what we call phage therapy. So I'd recommend, if you're interested in that, there's tons of cool articles and blogs online, take a look at that. And like I said, if anyone has any questions, just email me. I'd love to hear from any of you, or on Twitter. We'll try and answer as many as we can.
Thanks a ton, Sophie. Thanks a ton. I like you said, it was very high quality engagement, Sophie. The questions were seriously open questions in the field. And thank you for taking your time to answer them.
Yeah, I've taken down a few from my new grant proposals. So thank you, everyone.
All right, I know. Yes.
Thank you so much, everyone. And enjoy the rest of your day. It was lovely to meet you all. And best of luck with all your future scientific endeavors. I hope to see some of you in the field and at conferences when you're done doing your PhDs and stuff. It would be great to see you all.
Sounds good, Sophie. Sounds good. All right, Shreya, are we done? Yeah. All right. Thank you. Bye-bye, Sophie. Have a good day.
Thank you, everyone. Take care. Bye-bye.